African gorillas in the wild suffer from malaria. Like to humans, endangered great apes contract malaria. We now have new evidence from wild bonobos that the illness also hurts them. The deadly parasites that cause malaria are spread through the bites of infected mosquitoes. Although the initial signs of malaria infection in humans are mild, fever, headache, and chills, it can be lethal within 24 hours. There is little information available regarding the symptoms and lethality of malaria in apes. According to Emily Wroblewski, an assistant professor of biological anthropology in the School of Arts and Sciences at Washington University in St. Louis, we don't yet have a strong hold on the symptoms and mortality risk. There have only been a few number of infected animals in captivity that have displayed illness symptoms. They occasionally display signs like fever and other things that might be connected to infection, but not always. Also, it is quite challenging to track these things in the wild. Because researchers have found parasite DNA in the feces of wild chimpanzees and gorillas, scientists are aware that malaria infection is common throughout the geographic ranges of these primates. In fact, at least 12 different Plasmodium species have been found in African great apes, 7 of which are genetically linked to the human parasite that accounts for 95% of all human deaths. The bonobo, however, has so far avoided infection in all but 2 of the areas where scientists have studied them in the wild. Infected groups, where 38% of bonobos have parasite DNA identifiable in their feces, can be compared to uninfected populations from 10 additional sites throughout the native range in the Democratic Republic of Congo. The researchers can use this distinction to try to ascertain some fundamental details about how malaria affects the well-being and death of great apes. According to Wroblewski's latest study, which was released on February 23 in Nature Communications, bonobos are more likely to have specific immunity gene polymorphisms in regions where malaria infection has been identified, PAPA-B. The bonobo variants closely resemble a human variant, HLA-B*53, linked to resistance to the onset of serious, lethal diseases. This implies that these 2 animals may use similar immune defense systems. Given that these individuals are more likely to survive their infection, reproduce, and pass on the protective variants to their offspring, Wroblewski explained, to us, this suggests that there is a selective benefit to having these protective variants in individuals. The fact that infected groups differ from uninfected populations in this immunological feature raises the possibility that the virus may be harming bonobos' ability to reproduce or increasing their mortality, according to the researcher. The disparities across bonobo groups offer the first concrete proof, albeit circumstantial, that an infection in a wild great ape might have negative effects. Protection strategy. Beatrice Hahn from the University of Pennsylvania, who worked with Wroblewski on the paper, has tracked the trends in great ape malaria infection over the past few decades. Her earlier research contributed to the discovery that the most dangerous human malarial pathogen originated from a gorilla jump. To predict future transmissions, it is essential to understand the natural history and patterns of malaria transmission in our nearest relatives, according to Hahn. Due to the fact that the DNA utilized for sequencing was taken from feces that were collected after they were deposited, this research avoided handling or other disturbance of wild bonobos. The immunogenetic pattern found in infected bonobos is very similar to that found in human populations suffering from malaria in Africa, which piqued the scientists' interest. This is noteworthy because immune genes evolve quickly in an effort to keep up with rapidly evolving infections, according to Wroblewski. As a result, it is quite unusual to spot a pattern that both humans and their closest living ancestors share. She argued that further research should be done to determine how this specific immune gene variant protects people, both bonobos and humans, because knowing how it works can help develop new treatments or vaccines for people. Because apes are an endangered species, it's also crucial to comprehend how diseases are impacting them. Given the increased pressure from human activities, disease is a constant threat to their survival. I find it remarkable that both humans and bonobos respond to their infection in the same way, Wroblewski said. With the numerous ways in which each host could adapt in response to their infection.